Thank you very much, Dr. Kuama. It's very my great pleasure to present our data here. And also, thank you very much for coming this seminar. I'm very excited to see many people are coming. As Dr. Kuama introduced, I will show some data about stomach cancers, the unique feature of the stomach cancer in Japanese or East Asian populations. But as Dr. Mito said, that today we have 3 sessions related to cancer. I will briefly introduce the background of what is the cancer genome and how we will analyze these data today. Okay, so I have no COI on this page. First of all, I will talk about gastric cancers. Dr. Kuama said that the gastric cancer is more frequent in the East Asian compared to the Western countries, including US. This picture shows the frequency of the gastric cancer around the world. And as you see, the East Asia includes Japan, China, Korea, and some Asian countries, a very high frequency of the gastric cancers. But also you see that same frequencies observed in South Africa, South America, and also a part of Europe. So as you see that there is some the difference or diversity of the frequency in the world. So we don't know why. So we would like to know what is the cause of this kind of different frequency of this cancer. And also, as Dr. Kuama said, that the gastric cancer is very important cancer for our country. So we are starting to see cancer. So of course, there's many studies cover that there is some risk factors related to gastric cancer, including genetic factors, genetic variants, including some specific genes, or some environment factors, including the Helicobacter pylori infections or EB virus. So part of the gastric cancer is related to the infections. So that is also very unique feature of this cancer. But also there is other environmental factors, including smoking, alcohol intake, or some obesity reported to be associated with incidence of this cancer. So this is just the background of gastric cancers. So as I said, Helicobacter pylori is a very important risk factor of gastric cancer. It's a very tiny bacteria living in the stomach, but they stimulate inflammations in the stomach gastric epitheliums. And this kind of chronic infections is precancerous regions, which promote to the cancer by several steps. This scheme shows that the infection causes some coronary gastritis. It's a kind of inflammations. And after this kind of inflammation, the gastric epithelium started to regenerate. And then sometimes they cause some abnormal cells, and this proceeds to the cancers. So this is distribution of Helicobacter pylori infection. And as you see, this is very similar to the frequency of the gastric cancers. So that's why Helicobacter pylori infection is very important risk factor of gastric cancers. And also, in the pathological point of view, there is 2 histological subtypes of gastric cancers. One is intestinal type. It is a cohesive, is a type tumor cells associate very tightly, and this is called cohesive or intestinal types. And this type is major type of gastric cancer and highly associated with Helicobacter pylori infections. But also there's another important subtype, which is poorly cohesive or diffuse type gastric cancer. They are not much associated with each other, but the problem is this diffuse type is poorer prognosis and unclear with cause. So we don't know why this cancer occurs, but the problem is they are very dismal diseases. So that is the background of gastric cancers. And also, I would like to present some background of the cancer genomes. As you know, that cancer is caused by accumulation of many mutations in the genomes. So after the fertilizations, there is accumulation of mutations, somatic mutations in some cells. And when the mutation hits important genes, they gonna study to the cancers. So there is several mechanisms causing these mutations. One is very starting in the start of the birth. It's a germline variations, and also some environment factors are also very important for in causing the mutations. So, so how many mutations does the cancer have? As Dr. Kwama said, we have now new technologies to sequencing genes rapidly and cheaply. So this kind of, we can sequence a very large number of cancer genomes. So now we can profile the mutation data of many different types of cancers. So we can catalog these data, and this is the number of the somatic mutations of each cancer genomes. As you see that, for example, leukemia sarcoma is a kind of pediatric cancer, is a less number of somatic mutations, but this, for example, skin cancer or lung cancer or colon cancer has more mutations compared to these common cancers like breast cancer or gastric cancers. So on average, about 10,000 or 100,000 somatic mutations happen in each cancer genomes. That's a number. But the data says that not all of 10,000 mutations are important. So there are two types of mutations. One is driver mutation, and the other is passenger mutations. So this is depending on how these mutations are associated with carcinogenesis. So driver mutation is that is important for cancer initiation or progression. So that is very important genes. By contrast, passenger mutation is not important. Just happened by accident or some kind of factors, environmental factors, but they don't contribute to the cancer. So this is by the driving cars. The drivers is driving cars, but passenger just sitting in the car. It's like a cancer. So there's two types. And also we can know how many driver mutations does cancer have. So this is also based on the large number of cancer genomes sequenced. So on average, about 5 driver mutations are required for cancer. So as I said, 10,000 mutations happens, but only 5 is important. So it's a very rare event, but this number of genes, this driver gene is very important for cancer initiation and progressions. So I'm sorry, so we have more background. So that is associated with my studies because cancer genome data can tell us what is the driver genes, but also this data can tell how this mutation happens. Because as you know that there is various factors, environmental factors causing somatic mutations, but interestingly, for example, smoking or UV exposure of some carcinogens, they cause these mutations, but different place. That's why we can classify the somatic mutation based on its sequence context. Then we can tell which factors is inducing each mutations. That is called the mutation signature. So mutation pattern or mutation signature is associated with specific cause of cancers. That's why if this is some examples of signature, this is smoking signature. This is caused by the smoking carcinogens. This is predominantly C2A substitutions. And this is a UV signature. This is exposure to the UV. And they have different context, this is C2T substitution is dominantly. So based on this kind of different patterns, we can tell, for example, if you sequence some cancers, this cancer is caused by the smoking, or this cancer is caused by UV exposures. So using this cancer genome data, we can speculate what mutagenesis process works in each cancer. So here we have more than 50 mutation signatures identified in human cancer. So that's why more than 50 different mutation process or cause of cancer is working in a human cancer. And interestingly, these, as I said, each mutation signature linked to the specific cause. For example, aging or some lifestyle, including smoking or UV exposure or DNA repair deficiency or some chemical exposure or even in a treatment. So for example, chemotherapy causing this kind of specific mutation signature. So, so my analysis is uncovering cancer caused by the analysis of cancer genome. So if this is one example, if cancer genome is like a painting, so if then we can deconstruct the painting with colors by some mathematical analysis, and then each color is linked to the specific cause of cancer. So that's why if you see the picture and then you can deconstruct with colors, then you can tell which color is used in this picture and each color linked to the specific cause. That's why we can speculate what is the cause of this cancer. That is processing of the data, and I will present an example of how we can use this analysis for the gastric cancer. So, so we move to our analysis. So we have we sequence about 1,400 cases of gastric cancer from different parts of the world, and then we deconstructed the signature. So in gastric cancer, we have 14 different patterns happening. And we are very interested in this specific one, signature 16, I'll tell you later. And also, because we collected samples from different parts of the world, we can compare the difference between the ethnicities, for example. So this is data about if you compare to the European ancestry and the East Asian ancestry, you see some signatures, some patterns are more frequent in some populations, like signature 16 is more frequent in the East Asian population. So there is some difference of the frequency of a specific signature in different in each ethnicities. And also, as I said, there are two historical subtypes, cohesive intestinal and non-cohesive diffuse type. And also, this signature 16 is more frequently detected in the diffuse type gastric cancer. So each two gastric cancer have different mutagenesis processes. So next, I try to understand what is a signature 16 is associated with. And so I have to skip some part. Actually, this signature is associated with East Asian population and diffuse type of the gastric cancer. So what is the signature 16? That is more associated with alcohol intake, actually. And also, as you know, that there is two enzymes which metabolize alcohol. So ethanol is metabolized to acetaldehyde and then to acetate. There are two enzymes. And also, so germline variants of these two genes associated with how we can metabolize alcohol. And interestingly, the weak allele, which does not work very well about metabolizing alcohol, is more frequently detected in the East Asian populations. As you see, this green one is a weak allele of this enzyme. And interestingly, in Japan and China and East Asian populations, more frequent of this weak allele of this enzyme. So that's why this causes flushing. It's Asian flushing, is called so-called Asian flushing. And also this signature 16 is associated with this.
Is it associated with very expensive alcohol? Your pictures have very expensive.
Yeah, that's why I choose it. Yeah, I know. It's a very, I know, yeah. You know very well. Yeah. Romani Conti, yeah. Yes. And this signature is also associated with liver cancer and esophageal cancers. And next, we would like to know this signature is rarely caused driver mutation in gastric cancer. So we compare to the pattern of mutation signature and pattern of somatic mutation of the specific driver genes. And then we found that this signature 16 is very highly similar to the low-A driver mutations. So what is the low-A? Low-A is an important driver genes of diffuse type gastric cancer. That's very consistent with our data because we saw that signature 16 is more frequent in diffuse type, and also signature 16 is very similar to the low-A. It's a driver mutation of the diffuse type gastric cancer mutations. And also we verified that this specific low-A mutation was enlisted in the East Asian ancestry, diffuse type gastric cancer, and germline variants, which does not work well to metabolize alcohol. That's why our hypothesis is that if you have weak allele of the germline, which does not work well with alcohol metabolite, which is more frequent in East Asian populations, and if you drink a lot, then signature 16 happens, and then that causes specific driver mutations, low-A mutations, and that is an important key driver genes of diffuse type. That's why causing diffuse genes. That is our hypothesis, and this hypothesis is totally dependent on the data-driven. So we don't know any pre-hypothesis, but our data suggests that this kind of specific process is associated with specific frequency of the diffuse type gastric cancer in East Asia populations. So this data was published last year, and interestingly, NBC News picked up this data. And also, as you see that it is very important to compare the different ethnicity backgrounds of cancer genomes. So that's why we also started the international collaboration to uncover mutation signature around the world. This is called the MutaGraphs project, led by the Sanger Center in UK, and also IARC, WHO teams. And this project is supported by the Cancer Grant Challenge, which is Cancer Research UK, and NIH support this project. And this MutaGraphs expanded cancer types. I showed the gastric cancer today, but this project covers many different types of cancers, including the esophageal cancers, or kidney cancers, or pancreatic cancers, colorectal cancers. And Japan also contributed to this project. And as you see, that this is high incidence region, that this is low incidence region and Japan is high frequency of pancreatic cancer and colorectal cancers, but low incidence of gastric kidney cancer. So this project collected different cancer genomes for different regions of the world, and then we compared to the mutation signatures as presented. This is one example. So we sequenced esophageal cancer from different parts of Japan, different parts of the world, and also again we found that specific this signature 16 was also detected in the Japanese esophageal cancer and also in Brazil, sample from Brazil. So that's also again tells us that alcohol is a very important risk factor of this cancer. So this is a summary of my presentations. First, I showed some background of the cancer genomes and mutation signatures, and we applied this technique to the gastric cancer, and we found that signature 16 was associated with East Asian ethnicities, alcohol consumptions, and germline variations and diffuse type gastric cancer. And also, the by comparing the mutation signature and the specific somatic mutation patterns, we found that signature 16 contributes to the acquisition of low-A driver mutations. So this is one example, but if we expand this study from different country, different cancer type, different regions, we can covering and understand the diversity of cancer frequency based on the genome data analysis. So this is acknowledgement. This is done by the National Cancer Center and the IMSUT teams of this analysis. But I also thank to the cancer patients who agreed to use their samples for our analysis. And also, we have many collaborators for sampling and data analysis from Japan and Singapore teams. Okay, so that's my presentation. Thank you very much.
Thank you, Dr. Shibata. I think we have time for a few questions.
Yeah.
Fascinating study.
Thank you.
Fantastic example of germline polymorphism affecting somatic cancer mutations. I had one question. How did you assign more than one mutation signature per case, or did you just score the dominant signature in each case?
Actually, so we assigned many signatures from one cancer genomes, but we use a contribution of each signatures as a readout of the samples. And then we compare to the different ethnicities or different tumor types based on the contribution of each signatures.
So in those cancers, signature 16 was the dominant signature.
That's a good question. Actually, not. Signature 1 is dominant. It's aging-associated ones. But signature 16 is very unique or more frequent in a specific subtype and a specific ethnic background.
Mm-hmm. Yeah. Thank you.
Thank you very much. Yeah.
The germline variant RS641 is also known associated with esophagus cancer.
Right. Yeah.
Yeah. Are there any overlap between the population of the esophagus cancer and gastric cancer?
Yeah, that's good. Good question. Actually, so this I shows in my analysis. We focus on esophageal squamous cancers, and this type of cancer is more frequent in East Asian populations as in gastric cancer. So yeah, alcohol is very important risk factor of this esophageal squamous carcinoma, and that is consistent with gastric cancers also. Thank you. Thank you very much. Yes?
Thank you for very interesting presentation. So you mentioned we need 5 driver mutations in average.
On average, yeah.
Yeah. Is there any other genes that are associated with Rho A? I mean, so except for Rho A, so any other gene that's highly found in Rho A mutation, like a combination?
Okay. You mean a diffuse type?
Yes.
Gastric cancers. That's very good questions. Actually, I don't show in detail about the driver mutation of the gastric cancers. And actually, TP53 is, of course, very important driver genes. And this gene is also mutated in the diffuse type gastric cancers. And it depends on the samples. But we also observed that some receptor tyrosine kinase signaling, like MET, is one of the receptor tyrosine kinase, or some HER2. HER2 is not so much frequent. Interestingly, KRAS mutation is not so much frequent. But yeah, we do see some cases, yes.
Yes, sure.
Thank you so much for the exciting talk. Can I ask about the relationship between pylori and also the gastric cancer? Why this pylori? How this can develop the cancer?
Okay, thank you. Yeah, that is also a very important question. Actually, there is two concepts. One is, as I said, that pylori infection causes chronic inflammations, which stimulates regeneration of the gastric epitheliums, and then studied to the cancer initiations. But other studies like CAG-A, which is a very important bacterial proteins, and CAG-A is injected in the gastric epitheliums, and then it stimulates some signaling like receptor tyrosine kinase signaling, and also they causes the stimulation of the proliferation of the epithelial cells. And interestingly, Helicobacter pylori infection also inactivate the BRCA pathways. That's a very recent studies, and says that Helicobacter pylori infected gastric cancer has more BRCA associated mutation signature. So there is several pathways, bacterial proteins manipulate or interfere in the gastric epithelium. That is another aspect of the pylori infection in the gastric cancer initiations.
Any other questions?
Thanks for the beautiful talk. My name is Tatsuhiro Kumasaga from IMSUT. I have a question about the smoking signature. So I think in Japanese population, there is a very common germline deletion at CYP2A6 locus, over 20% in the frequency. So is there any some interplay between this germline deletion and signature somatic mutation?
I see. That is, thank you very much. That is also very interesting. I didn't analyze smoking-associated cancer like lung cancer or other cancer, but we didn't check about the association between the specific germline variants and this signature. And interestingly, this smoking signature is also observed in liver cancer. So that's why this is also very important cancer risks. But as far as I know, there is no specific studies focused on specific germline variants associated with specific signature or frequency of somatic driver mutations of this cancer type.
Thank you very much.
Thank you very much. That's very interesting.
So maybe we need to move on.
Sure.
But an anecdotal experience of mine, my grandfather died of esophageal cancer after having drunk a big bottle of sake every day for years and years. And then your talk was about signature 16, which is affected by alcohol and which is related to esophageal cancer. So it's sake does not seem to be just stomach cancer. So if you focus on one reason, one driver damage, which may be able to explain many types of different organs cancer.
Right.
Yeah.
I don't show data today, but the liver cancer is also associated with
Yeah, so liver, esophagus, and the liver cancer is quite often caused by virus in Japan. But maybe sake, anybody who has chronic hepatitis, drinking sake, they very high frequency of hepatocellular cancer. So thank you very much for your interesting finding. And I think I just would like to add Yamazaki, very expensive Japanese whiskey, usually does not give me headaches. So probably that will not cause that much of cancer as the other cheap whiskey. That's what I would like to. Huh?
What you tell yourself.
Yeah. So the thing is, I would like to enjoy Yamazaki. So great. Thank you very much. So thank you very much, Professor Shibata.
Yeah.